Hello students, welcome to the EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, a Central University, New Delhi. Today I am going to talk about the module ultracentrifugation from the paper Centrifugation Technique. This module we discuss the following thing in detail. First we discuss about the basic concept of ultracentrifugation. What are the basic concept of the ultracentrifugation? How this technique is more advanced to the other form of the centrifugation. Then the types of ultracentrifuge. There are two types of the ultracentrifuge. One is the preparative and one is the analytical based on their application. So there are two types of ultracentrifugation. We discuss a brief introduction about both these form of ultracentrifuge. Then we discuss about the type of rotors for the ultracentrifuge, because the rotors used in the ultracentrifugation are different from the normal centrifugation and they have some advanced feature, of course. Then we discuss about the principle of ultracentrifugation techniques based on the principle of sedimentation velocity and the sedimentation equilibrium. And at last we discuss about the application of the ultracentrifugation techniques. So the ultracentrifuge is actually the most advanced form of the centrifuge, and it is a very costly techniques. It is actually the additional advancement, additional features applied to the normal centrifuge, and these features are in terms of the speed. So here you can run up to the more than 100,000 of G value, and that is the mainly helpful for the separation of many molecules, biological molecules, from each other. It having both application like the preparative and analytical. So before that I will give the idea that it is precisely used for the determination of sedimentation coefficient, which actually measures the size of the particle, molecular weight of the molecule, molecular shape of the molecule, and molecular interactions such as protein-protein interaction, protein-ligand interaction, protein-DNA interaction. All kind of interactions can also be measured using the ultracentrifuge. So it actually works at the very high speed, or RCF. That is the main advanced feature. The second thing is that here people use a small volume of the sample, that is the less than 1 ml, to precisely measure the differences or the separation properties of the molecules. Then it may uses relatively pure sample. If you go for the analytical purpose, people prefer to use the pure sample, and if you want to use for the separation, then some impure sample can be used for the fractionation of the cellular components, as in the case of preparative centrifugation. The analytical centrifugation having inbuilt optical system to analyze the movement of molecules during the centrifugation process, and it runs at a speed of more than 1 lakh G. The one very important thing with this ultracentrifuge is the application of the vacuum system, because this runs in the chamber where the pressure is 0, means it is a vacuum. If there is a pressure, then the air creates a frictional force, and the frictional force is so high that the chamber can burn out. So to avoid such kind of friction, the chamber should be vacuumed, so there is no frictional forces, and the centrifugation can be done very easily. This can be used to palette the membrane, ribosomes, and other cellular organelles. They use in the preparative or both in the analytical, the gradient systems are used, like the cesium chloride gradient for the separation of the DNA, and the sucrose density gradient used for the paletting cell fractions, small proteins, ribosomes, etc. Here we are going to discuss about the preparative ultracentrifugation techniques. Preparative ultracentrifugation technique is utilized for the separation of complex biological mixture, and that mixture can be further utilized for the another purposes. It can use to fractionate the fine cellular components such as mitochondria, and other cell organelle from a mixture of complex organelle system. It is also used for the purification of proteins, nucleic acids, and plasmids. Moreover, it also utilized for the characterization of the molecular interaction. That is the main application of the preparative ultracentrifugation techniques. Here you can see the analytical ultracentrifugation techniques. Analytical ultracentrifugation technique is as similar to the preparative ultracentrifugation technique, but the major difference between these two techniques, there is further advancement in the preparative centrifugation technique, that the addition of some detector that could easily identify the what's process going inside the centrifugation. It is widely used for the determination of hydrodynamic properties or thermodynamic properties of biomolecules in the absence of solid support, like earlier people used for the electrophoresis and chromatography. Here you don't need such kind of support during the ultracentrifugation techniques. It used to measure the relative molecular weight of a substance, either protein, DNA, and any biological molecules. Moreover, it also utilized for the molecular shape determination, aggregation behavior of proteins or any biomolecules. For example, it is either dissociating or associating with respect to some certain changes or certain condition that can be determined through the analytical ultracentrifugation techniques. It also used for the protein-protein interaction measurement, that what are the how many proteins interacts with how many proteins, like the stoichiometry of the protein as well as the binding affinity of the protein with ligand or protein-protein interaction. So these are the major application of the analytical ultracentrifugation techniques. Now, analytical ultracentrifugation is based on the two major component or major principle. One is the sedimentation equilibrium and the another is the sedimentation velocity. So the sedimentation equilibrium method is utilized for the measurement of thermodynamic properties, measurement of absolute molecular weight, but it is the shape independent, and it can be utilized for the aggregation analysis as well as protein-protein interaction. However, sedimentation velocity is mainly based on the hydrodynamic properties, and it is used for the relative molecular weight of the substance as compared to the in sedimentation equilibrium it is used for the absolute molecular mass measurement. Here in the sedimentation velocity you can measure the molecular shape also, because it utilizes the shape in also consideration for the calculation, and of course it is utilized for the determination of aggregation behavior. You can see here that the centrifugation techniques and especially the ultracentrifugation technique is based on the Svedberg equation, and the Svedberg equation is a simple mathematical derivation of the centrifugal force. You can see if you rotate a substance in a centrifuge, there are many forces acting on it. The first is the frictional force, then buoyant force, and then centrifugal force. So the centrifugal force is equals to frictional force and the buoyant force. So the total force acting on a particle is given by an equation, like the centrifugal force equals to F equals to m omega square r, and the buoyant force equals to m omega square r with m dispersion, and the frictional force is proportional to the frictional coefficient into the V. So this equation is actually utilized for the calculation of properties of the macromolecules during the process of ultracentrifugation, and this is given it will reach a constant velocity where the forces balances each other. You can see here in the equation that m omega square r minus m dispersion omega square r minus fv equals to 0. So it means that these two forces, buoyant forces and frictional forces, counterbalance the centrifugal forces, and that is the basic principle of the ultracentrifugation technology. Sedimentation of particles in the gravitational field is dependent on the mass of the particle or the density of particle or the size of the particle, but it is better explained in terms of sedimentation coefficient, that is known as S. So S value is the sedimentation velocity of the molecule. This may be measured by sucrose density gradient sedimentation or by the analytical ultracentrifugation. The S value of molecule is determined both by its shape and its mass. So, it is a very important factor for the calculation of the biophysical properties of the micromolecules. It is the best described in terms of mathematical expression, and it is given as the sedimentation coefficient S equals to mu divided by the omega square r, where mu is the velocity of the particle and omega is the angular velocity of the rotor and r is the distance from the axis of rotation. So, thus S value is related to the molecular weight of the solute, and S equals to molecular weight into 1 minus V rho divided by NF. This is the another way to calculate the in terms of the molecular weight. A correlation should be established between the molecular weight and the S value. It is given as S equals to a molecular weight into 1 minus V rho divided by NF, where V is the partial specific volume of the solute and rho is the density of the solvent. N is the Avogadro number, that is 6.023 into 10 power minus 10 to power 23, and F is the frictional coefficient of the solute. But you must be sure that these factors are dependent on the temperature. So, the temperature should be taken in consideration, and at 20 degrees Celsius it is considered that the sedimentation coefficient value is the 10 to the power minus 13 second is called as 1 Svedberg unit, because this term was first time given by Svedberg in 1940. So, it is given by the symbol of S, and that implies in the unit of S per radian square, but the radian square is generally ignored. So, only the S is taken in consideration for the measurement of physical or biophysical properties of macromolecules. So, velocity of the separating molecule is determined actually on the partial specific volume of the solute, and its unit is the unit of partial specific volume is given as centimeter cube per gram. So, the partial specific volume of a protein is the ratio between its volume and the molecular weight. The partial specific volume of molecule is a measure of change in volume of the solution per gram of the molecule in that solution, and the partial specific volume is equal to the inverse of the density of the particle. So, V can be measured directly or estimated. So, it is a very important parameter that has to be considered during the ultracentrifugation process. So, you know that how the partial specific volume of protein is calculated. So, here we are discussing how, if you have given a sequence, then how you can calculate the partial specific volume of a protein molecule. If you know the constituent amino acid, it is given by the equation equals to V equals to sum of Ni, Mi, Vi divided by sum of Ni, Mi, where N is the number of residue of I type and M is the mass of that residue or the mass of I type residue, and V is the partial specific volume of I type residue. So, these parameters can be used to calculate the partial specific volume of a protein molecule or any substance. However, you must be aware that the estimate breaks down if you have detergent, bond modification, etc. And the molecular weight of amino acid minus 18 is 18 always deducted, because the amino acid in a protein, once it forms the peptide bond, the one water molecule release per peptide bonds, that's why 18 has to be deducted. So, these are the parameter used for the calculation of partial specific volume of a molecule. So, here I am giving you one example for the calculation of partial specific volume of a solute. For example, one rough estimation of partial specific volume of a protein which may be used if the sequence of protein is not known. So, the average partial volume of a protein is 0.725 centimeter cube per gram. Because the average of experimentally determined partial specific volume for soluble globular protein is approximately 0.73 centimeter cube per gram. Average of experimental value of 13 soluble proteins This values varies from protein to protein, but the range is rather narrow because either 0.7 or 0.75. That's why we can take 0.725 as an average for the calculation of partial specific volume of protein. Then we also know the density of the solvent because it is equally important parameter to understand, and you know the density is given as gram per centimeter cube. So the density of many buffers may be approximately equal to the density of water. So the density of water is 0.998 gram per centimeter cube at 20 degrees Celsius. However, if you use the different kind of buffers, then you have to take that composition of that buffer, and especially the molarity of the solution is also an important factor for the taking consideration of the density of the solvent. One software known as Ultrascan has a feature for calculating density of common buffers with common additives, and that can be utilized for the data analysis or the ultracentrifugation techniques. Another important feature is the frictional coefficient because it has a great impact on the biological separation, in the separation of molecules, especially the biological molecules. So the frictional coefficient is a very important factor, and it is given by the Stokes equation, that is the F equals to A into 6 pi eta, where A is the Stokes radius, approximately the length of long axis of the molecule. The Stokes radius can be determined by gel filtration, and eta is the viscosity of the solvent. So these two properties actually measures the frictional coefficient of a molecule. So the bigger the size, they have the higher the frictional coefficient. In the same way, if the viscosity of the solvent is very high, then it has a high frictional coefficient. If the viscosity of the medium is low, then the frictional coefficient is low. So before understanding the ultracentrifugation technique, you must be aware with all these properties of the molecules that are taken in consideration during the ultracentrifugation techniques. So now there are the sedimentation coefficient value of some well-known biological molecules are given here. So these are some particles like the lysosomes, which having the sedimentation coefficient value is 19400s, tobacco mosaic virus having 198s, the ribosome is having sedimentation coefficient value 80s, then ribosomal RNA molecule having 20s, and transfer RNA molecule having 4s. On the other hand, the protein like the hemoglobin having only 4.5s. So the known sedimentation coefficient values of some of the biological molecules that has been utilized for the determination of the ultracentrifugal properties of molecules using ultracentrifugation techniques. So you can see here in this figure the structure of ultracentrifuge. It's a very high-end equipment, and the samples are loaded into the special centrifuge cells with transparent window for the optical measurement, and the distribution of solute molecule during the experiment is monitored by the optical system. The cells are scanned across the entire radius and data automatically collected for the subsequent analysis. Now we will discuss in detail about each component of the ultracentrifugation equipment. Rotors are the first and most important component of the ultracentrifuge, and this rotor is typically different from the other rotors of the common centrifuge. It is a very highly massive and that stores kinetic energy. It comes in the two form, both as a fixed angle rotor in which the tubes held at about 45 degree angle to the vertical, and the swinging bucket tube on hinges at full speed they do perpendicular to the gravity. The type of rotor one use is determined by the circumstantial experiments and the sample with the limitation of some particular type. Most rotor have opening to place sample tube of a specific size and volume. Fixed angle rotor embody the opening of the sample tubes lying at the fixed angle from the axis. Other rotor enclose openings perpendicular to the near to vertical sample tubes. Swinging bucket rotor comprises a ring of individual tube holder at the hinge that helps the tubes to swing outward as the spinning begins. This results in the horizontal and alignment of tube with the direction of force. There are another rotor that is known as zonal rotor that are designed for the holding the sample of a greater volume instead of multiple sample tube, with the exception of these multi-sampling type of the rotor. Self-locking mechanism is yet another important feature for some rotor and making rotor installation trouble-free for working the safer. So these are the few important points has to be considered and should be known about the rotors of the ultracentrifugation. So now we will discuss about the design of this ultracentrifuge. You can see here that in analytical ultracentrifuge there is a method for detection is available with the optical detection system that measure the sample concentration inside the centrifuge and cells during and after the sedimentation. The centrifugation parameters and data acquisitions under the computer control, so you can see the realistic picture of the centrifuge tube on your screen. You can see here this is the rotor, this is the centrifuge part, and this is the cell which holds the sample, and there the cells having two parts, one is the reference and one is the sample component, and it is connected to the optical system which can detect the molecules inside the centrifuge tube during the centrifugation process. And in the case of analytical ultracentrifuge, a spins rotor at a controlled speed and constant temperature and records the concentration distribution of sample at known times. That depends on how much time you want and what you have fed it in the program. And the concentration is determined for solute obeying the simply Beer-Lambert law, that is A equals to ECl, where A is the absorbance, E is the molecular extinction coefficient, and L is the path length, by measuring the absorbance of the sample at a given wavelength at fixed position of the cell. So this diagram clearly show you the actual picture of the centrifugation, ultracentrifugation technique, and how the molecules got separated and how the optical system detects the actual phenomena going inside the chamber. In sedimentation velocity, there are two chambers, one for the sample and one for the reference, and cells having double sector centerpiece. SV cells are cylindrical and have double sector centerpiece. Figure is the top-down view of the cylindrical eyepiece. One sector is for the loading the sample and other is the reference sector that contains the solvent only. The reference sector is filled slightly more than the sample sector so that the reference meniscus does not obscure the sample profile. A uniformly dispersed sample solution is placed in the cell and sufficiently high angular velocity is applied to use rapid sedimentation of solute towards the cell at bottom. As a result, there is a depletion of solute near the meniscus, causing the characteristic spectrum like figure here you can see, and it reaches the plateau. A sharp boundary occurs between the depleted region and sedimenting the solute, as you can see here in the plateau formation once you plot the radius versus the absorbance. So here we talk about the design of optical system of ultracentrifuge. So the ultracentrifuge, if it is the preparative, they don't have any optical system, but if it is an analytical system, then there should be an optical system which used to take the actual picture, the dynamic picture of the molecule which is running during the centrifugation process in the ultracentrifuge. So the people used absorbance based optical system to measure the properties based on the absorbance at the particular wavelength, and the range is from 200 to 800 nanometer, and that is used for the detection of those molecules which having chromophore group that have tendency to absorb the light at particular wavelength. But some molecules which don't have any such property, that is devoid of such property to absorb the light, then in that case the Rayleigh interference optical system are used for the measurement of sample concentration, that is based on the refractive index changes rather than the absorbance. So it is used to analyze the macromolecules lacking the intense chromophore, for example polysaccharide, and samples that contains a strongly absorbing buffer component such as ATP, GTP, DTT. So these are the two optical system that is associated with the analytical ultracentrifugation technique for the measurement of dynamic picture or dynamic calculation of the for the measurement of molecular mass and other biological features of a particular molecule during the ultracentrifugation process. Now you can see here in the figure the velocity of individual particle in SV in the sedimentation velocity experiment cannot be resolved, but the rate of movement of boundary region can be measured. From this, the sedimentation coefficient can be determined. Remember, the sedimentation coefficient depends directly on the mass of the solid particle and inversely to the frictional coefficient, which is a measure of size of the solute particle. And that's why if you measure the sedimentation coefficient, then you can easily calculate the molecular mass of the substance that you want to know, or the mass of the substance of the interest. You can easily measure it. Now this another example of this is the example of sedimentation equilibrium. In the case of sedimentation equilibrium, there are the cells contain 6 columns, one row of 3 columns for the sample and other are 3 for the reference molecule. One row of the sample is sample row and one other row is reference row containing only solvent. As in the sedimentation equilibrium experiment, the reference column is fixed with more than one sample column. The solvent column, the concentration are varied usually from 0.25, 0.5 and 0.8 optical density at 280 nanometer, or same fraction of absorbance at any selected wavelength, where the wavelength will be monitored by the analytical ultracentrifuge. Also, several scans are taken at a given rotor speed to try to ascertain that the equilibrium has been reached or not. If it has been reached in scan 1, then you can take scan 2 an hour later and then look the same. So the sedimentation equilibrium experiments have a lower rotor speed than the sedimentation velocity experiment. Solute particle do not pallet at the bottom in the cell, but instead they process the diffusion, oppose the process of sedimentation until after a period of time. The two opposing forces reach equilibrium and the apparent concentration profile does not change. At equilibrium, the concentration of the solute increases exponentially towards the cell bottom. Each column displays a different absorbance profile because the concentration of sample are varied in each. You can see here in the picture a sample picture showing the sedimentation equilibrium experiment. Now we discuss about the equilibrium sedimentation. You can see here at the y-axis the concentration and the x-axis the radius. So the meniscus is formed at the initial condition at the cell bottom. So from the lower side the meniscus diffusion and the upper side is the sedimentation. So the diffusion occurs at the moderate speed. After sufficient time, an equilibrium is reached between sedimentation and diffusion, resulting in monotonic solute distribution across the cell. However, in the case of sedimentation, non-linear curve fitting can regularly determine the solution molecular weight, the association state, and the equilibrium constant for the complex formation. That is the major feature of the ultracentrifugation technique. Here you can see an example of the binding or association between the 2 or 3 molecules of the transferrin at a given concentration. So here the data for transferrin at 3 loading concentration are given here. At 3 data sets were fit simultaneously to monomer dimer equilibrium model, and the fit returned a Kd value of about 100 millimolar for the dimerization. A relatively small and randomly distribution residual indicates that the model proved a good fit for the database. So that is the major application for the ultracentrifugation technique, to calculate the binding affinity and the stoichiometry of the molecules in the solution. Now we discuss about the application of ultracentrifugation. As we already discussed the method and the principle and everything, now we discuss about the application of the ultracentrifuge. It is used for the examination of sample purity, that the quantitation of the size distribution in a polydispersed sample. For example, if you have any component biological like the protein, like carbohydrate, or any cellular organelle, you want to see that whether it is pure or there is some contamination. So that can be performed by using ultracentrifugation. It can be used for the calculation of molecular mass, and it is applicable for the large molecular mass range from 100 to 10,000 kilodalton. So it's having very wide range for the calculation of the molecular mass, and also it gives you the properties of the molecular mass in the solution form. Usually the other technique that is used for the calculation of molecular mass, that is not in its native form, like in the SDS or in the mass spectrometry. There are harsh condition, but here the condition is as good as the native condition. It also used for the analysis of associating system. For example, if you have two protein and they or one protein forming the oligomer with reference to their change in certain physical condition, then that can be determined by using ultracentrifugation. And it also used to analyze the stoichiometry, what is the ratio of two molecules, 1:1, 1:2, 2:1, like that. Then sedimentation and diffusion coefficient measures the macromolecular complex size and interaction, that how big the complex are formed, how in the same way, reverse way, how the complex are broken into the different component. This technique is also utilized for the ligand binding studies to distinguish ligand from the acceptor to differing absorbance. So I hope that you got a detailed idea about the centrifugation technique. So in summary, the ultracentrifuge is a special type of centrifuge which rotates at a very high speed than the standard centrifuge, and the typical rotation species in this centrifuge ranges from 30,000 to 50,000 rpm. And ultracentrifuge is usually used for separating macromolecules from solvent or for fractionating mixture of the molecules, and centrifuge are used for analytical as well as both the preparative purposes. The analytical ultracentrifuge is mainly used for studying the properties of molecules as well as analyzing the complex mixture of the molecule, like how many component of this mixture, what is the relative content of these mixture, and of course for the separation of these mixture. The preparative centrifuge is used for the macromolecules such as protein, nucleic acid, based on their physical properties, and in the preparative centrifugation the sample is further utilized for the biological purpose. However, in the analytical centrifugation it is used for the measurement of the physical properties of the particular macromolecule or the substance. So this is the end of this module, and thank you very much.